X-Talks connects professionals in the life science, medical device, and food industries with useful content like webinars, job openings, articles, and virtual meetings to help you succeed in your career. This life science-focused podcast brings together some of our editorial staff to share insights into the latest B2B industry news to keep you up to date. X-Talks is excited to introduce the inaugural issue of X-Talks Clinical Edge, where innovation meets expertise. This digital magazine offers you exclusive conversations with some of the brightest minds and trendsetters of the clinical trials community, offering you a front row seat to the evolving landscape of clinical research. As you dive into the rich content of X-Talks Clinical Edge magazine's first issue, you'll discover insights from pharmaceutical companies, patient advocacy groups, leading clinical research organizations, and an ensemble of key opinion leaders from organizations like the Clinical Trials Transformation Initiative and the Mayo Clinic. Follow the link in the show notes or visit clinicaledge.xtalks.com/issue 1 to dive into the first issue of X-Talks Clinical Edge magazine and be a part of the conversation shaping the next era of clinical trials. That's clinicaledge.xtalks.com/issue 1.
Hello and welcome to the X-Talks Life Science Podcast. I'm Aisha Rashid, Senior Life Science Journalist at xtalks.com, and this week I'm delighted to introduce a very special guest on the show. Today we have with us Dr. Adam Kaplin, who is the co-founder, president, and chief scientific officer at MIRA Pharmaceuticals. MIRA Pharmaceuticals is a preclinical stage pharmaceutical company focused on the development and commercialization of new molecular synthetic cannabinoid analogs for the treatment of adult patients with anxiety and cognitive decline, typically associated with early-stage dementia. This patient population also typically experiences neuropathic pain. The company's initial drug candidate, MIRA-1A, could be a significant advancement in treating neuropsychiatric, inflammatory, and neurologic diseases and disorders. The company also has a second drug candidate, Ketamir-2, which we will also learn more about in the show. Dr. Kaplin is a neuropsychiatrist whose research focuses on the investigation of the biological basis of immune-mediated depression and cognitive impairment by using multiple sclerosis as the model. Dr. Kaplin transitioned into industry by joining MIRA Pharmaceuticals to help lead drug development in the space. Prior to joining MIRA Pharmaceuticals, Dr. Kaplin served in a number of positions at Johns Hopkins University, including Principal Neuro-Psychiatric Consultant to the Johns Hopkins Multiple Sclerosis Center of Excellence, among others. Dr. Kaplin also served as co-founder of numerous healthcare-related startups, including from 2018 to present Reward Pathways, which is a company devoted to addiction treatment focused on a combined eHealth and medicine approach. Dr. Kaplin's original research has been published over 70 times in peer-reviewed publications in key areas in neuroscience and immunology, with a particular focus on the intersection of neurology and psychiatry. He has also authored or co-authored numerous review articles and textbooks. To learn more about drug development in the space of dementia, cognitive decline, and anxiety, and to specifically learn more about MIRA Pharmaceuticals' drug development programs for these conditions, let's turn to Dr. Kaplin. Dr. Kaplin, thank you very much for being on the show today.
Thank you for your kind words and the chance to, sort of get the word out about what we're working on. So I look forward to talking to you the issue about these things.
Likewise, I look forward to learning more about MIRA Pharmaceuticals and your work in the space. I'd like to start off by asking you more about MIRA Pharmaceuticals in terms of how and when it was founded and what its core missions and goals are.
So we've been working on the various the drugs that you have mentioned, MIRA-1A, and I'm just going to call it Ketamir. There is a 1 and a 2, but the 1 is probably not going to ever be developed because of the advantages of 2. So I'll just refer to it as Ketamir and MIRA-1A. So we've been working on these drugs for at least the past couple of years with MIRA-1A and probably 6 months for Ketamir. And the company did have a successful IPO back in August of 2023, so just a few months ago. and we've been working hard on trying to sort of find our way on a timeline to work as quickly as we can to develop these drugs and see if we can bring them forward and ultimately see if they're successful in being able to address the needs that I know we'll get into related to dementia, early stage dementia, and treatment-resistant depression. So.
And coming from an academic background, it's very interesting to see professionals and leaders like yourself moving from academia to industry. And so, how has that transition been like? And what led you to join specifically MIRA?
Yeah, so I mean, it's a great question. It's interesting because being a psychiatrist, I certainly know what it's like for stigma for my patients. The interesting thing has been that the stigma that exists between academicians, some of them, and industry. So I have colleagues who will cross the street rather than talk to me anymore. Other colleagues are
Oh no.
quite interested in this, but the reason is really quite simple. I think that just qualitatively, all of the really exciting new ideas, not all of them, but the majority of them come from academia. In academia, we still have the ability to sort of think outside of the box, do some of the, Hail Mary experiments that are, high chance of failure, but high chance of making not just incremental, but revolutionary changes. That's harder to do in industry because in industry, there's always, when you're spending over $1 billion a drug, you don't want to take chances on things that have, less of a, considered, carefully weighed risk benefit kind of thing. But on the other hand, in academia, we are not very good at taking these ideas, translating them into treatments that we can then use to help millions of patients. And if we're not part of the discussion as academicians, so the mission that I have as an academician is, my purpose in life, if you will, is to really try to help change the lives of people suffering from mental illness, particularly depression and cognitive impairment. And if we're not part of that discussion in industry, we won't be able to have a say in how things go. Just quantitatively, there are roughly 50 million publications that come out each year, and yet we had,
Wow.
I think it was something on the order of 40 FDA-approved treatments last year. So, all of those publications are great, and many of them make significant and dramatic contributions, but clearly they don't translate well into actually changing things. So the estimations are that there are, let's just ballpark it, 10,000 different diseases out there. We have treatments for 500 of them. And so we are greatly in need of people to sort of be willing to bestride this great divide between the people who know how to make drugs in industry and the people who know, new ideas and what the real clinical needs are from working with patients. So long-winded way of saying that my hope is to convince other people that this is the combination of academia and industry is really the way we're going to make most progress.
Much needed. Thank you so much for those insights. Wow, I didn't know the discrepancies were that stark there. Very interesting. And so, Dr. Kaplin, in terms of depression and cognitive decline, what are really the unmet needs in the area and the gaps which you're hoping to address at MIRA?
So with respect to depression, that's there are considerable needs. The needs, and I'm going to focus on the things that are related to ketamine. There are great needs that we have in addressing the continued rise in suicide rates, particularly among young people, where suicide is the second leading cause of death between the age of 10 and 34. I mean, 10th leading cause overall. This is 90% of the time due to depression. So there's great needs in a broad sort of way, but I think that one of the needs that is related to this is the fact that in the STAR-D trial, the largest randomized trial, multi-center trial looking at the treatment of patients with, if they didn't respond to one particular combination of treatment for their depression, they then went to the second, then the third, then the fourth. And even after 4 different treatments, 33% of people did not achieve remission. So, that's really demonstrating the difficulty that we have as a profession, which is how are we going to address the 1/3 of people who are in need of treatments that go beyond our conventional therapies. And yes, I would be remiss if I didn't mention that electroconvulsive therapy still is, done in its modern ways under using neuroanesthesia, a safe treatment that can be done and have rapid effects, but the side effects are cognitive and the like. So we're good at delivering electricity with ECT and transcranial magnetic stimulation, but many people don't want those types of treatments. They're too interventional and have too many side effects. So, the opportunity to try to work on treatments that will, one, treat depression for that 1/3 of people who don't respond to multiple trials. 2, our best treatments still require 1 to 2 weeks to even start to kick in, because you have to decrease. There's a break on the serotonin system called the 5-HT1A receptor. You have to downregulate that receptor before serotonin levels will actually go up. That takes 1 to 2 weeks. So, if you think about the absurdity that when we hospitalize patients, insurance companies will pay for, 4 days, 6 days of treatment, which is really before our drugs will even kick in. And we're sending people out. So having more rapid treatments is also a great need. And then the second great need is novel treatments. So, we've been doing the monoamine approach of norepinephrine, serotonin, and dopamine since the 50s. And, really having an ability to sort of broaden our armamentarium to other receptors, in this case, the NMDA receptor, with ketamine, and ketamine is an analog of that, are other needs. So with respect to MIRA 1A, everybody talks about the white tsunami, the aging of the baby boomer population, and how that we don't have as many young people. We're top heavy in terms of age distribution to be able to pay for and care for our aging population. And quite honestly, we've had great difficulties in targeting the right targets, I believe. I think many people agree that beta amyloid and these tangles are part of the puzzle, but certainly not the whole puzzle. And so coming up with treatments to help our aging population, where age is the biggest risk factor for dementia, is also a tremendous need. It is estimated that for every month that someone with Alzheimer's spends in a residential care facility, that's somewhere between $50,000 and $100,000 is what it costs. So you can think keeping people out of those facilities and well enough to live independently for a year has tremendous implications. So
indeed. And so you've mentioned, and we've touched upon, of course, the company's drug candidates, MIRA-1A or MIRA-1, and also ketamine. So I'd really, really love to learn more about these two compounds, how they came about as being sort of the front runners of your drug development program at MIRA, and your mechanism of action, what diseases they target, and the target patient populations, and all of that.
great. How long do you have? No, I'm just kidding. Let's see if we can cover, and please interrupt me and ask if there are questions. So, to begin with, THC, delta 9 THC, which is the intoxicating component of cannabis, marijuana, is, has its intoxicating effects at the CB1 receptor. So it turns out we all have within us two cannabinoid receptors, CB1 and CB2, that respond to, just like we have endogenous opiate receptors, we have endogenous cannabinoid receptors, CB1 being the one that is THC has its intoxicating effects, but also therapeutic effects. So THC essentially has three six-member rings in it and a tail. There are some, I'm just going to simplify for the purposes of simplicity. The furthest opposite end of the molecule from where that carbon, 5-carbon tail is a six-member ring that we just have taken a single carbon out and made into a 5-member ring. And that has made an enormous difference in terms of the activity of the drug. Now, let me just take one step back and answer well, why take that approach? Why not just, work on different delivery systems for THC, et cetera? And, THC, while absolutely having therapeutic effects, it is approved in a FDA approval, for instance, for dronabinol, which is synthetic THC, to treat things like nausea, chemotherapy-induced nausea, to treat things like decreased weight by increasing appetite and stuff. But really, when you take a step back and think about it, these drugs were really made by plants, in this case, cannabis, to try to prevent themselves from being eaten. So the plants upregulate the production of these drugs, just like nicotine and caffeine and the like, when they are stressed. We just happen to like these intoxicating effects, but they were designed by the plant to try to get predators not to eat them. So it's not surprising that they would have negative side effects. So in addition to the help that they provide many patients with pain and anxiety and sleep and nausea and the like, and I'm talking about medical marijuana now. I'm not trying to make claims for dronabinol that they don't have in their FDA approval insert. But the essential thing is that there are significant side effects, among them that THC is cognitively impairing. And we know that there are increased numbers of motor vehicle accidents. I can tell you that I've had a number of patients with MS who have turned to cannabis as a way of helping relieve their pain and the like, and they can't return to their former functions as lawyers or doctors or what have you, and they make a big trade-off. Interestingly, the studies that were done in MS to show the benefits for spasticity using THC or cannabis, all the patients had to, when they signed up for the study, had to agree not to drive during the 6 months that they were intoxicated. The problem is everybody wants the benefits, but not the burdens. So people say, oh, I want the pain control, but I don't want to not drive. And so, we know it affects people's cognition. And then people will sometimes say, well, but I only use it, on the weekends. So that's fine, but we should be able to come up with ways of controlling your pain, not just on the weekends. The other thing is they do increase heart rate for those people who are vulnerable that can cause heart attacks. And the main concern for many of us in the psychiatry area is that they absolutely do worsen bipolar disorder, make people more likely to have psychotic symptoms or become suicidal. And the data continues to come out, but it looks like THC, if you have a first-degree relative or certainly a family member who has schizophrenia, you are more likely to develop a chronic psychotic illness from consistent use in those situations. So for some people, it clearly isn't the right treatment. It does have side effect burdens in addition to the therapeutic effects. So we were really working on how do we make a better widget? How do we get a THC 2.0? So removing this one carbon has made a big difference. The first thing is it's no longer THC. That's a big deal because in reviewing it, because it doesn't go through any intermediates which are known to be addicting, cannabinoids or psychotropic drugs like THC. In reviewing the drug structure, the DEA has given us a assessment and said that we are not a controlled substance. Now, at the time that a new drug application is put in, obviously the FDA and DEA will have a chance to reassess that, but it really helps in the development process to not have this be a controlled substance. It's not a Schedule I drug like THC. So that's one of the initial benefits. But what had become sort of the main and surprising benefit, to be 100% honest, is that we found that in our studies that THC hits a high affinity site and a low affinity site. So essentially at very low doses, it hits a site, and this is when we looked at this in cells that expressed just CB1 receptor, and then looked at the second messenger system. So once that receptor is activated, it then leads to changes in cyclic AMP levels in neurons. In this case, we looked at, it was expressed in Chinese hamster ovary cells. And what was interesting is this low dose does hit this receptor that appears to have the therapeutic benefit. So that's where you see decreased anxiety. You do not see cognitive impairment, and you don't see weight gain as far as we have ascertained so far. And there is a site that at high doses with THC you also hit. That's where the response actually does an inverted U. So it sort of goes up in terms of the cyclic AMP response, and then it comes down. What fits it coming down is an inverse agonist. And the only reason I bring that up, getting a bit technical here, is that rimonabant was an inverse agonist. That was a cannabinoid drug that was approved in Europe back in the mid-2000s for weight loss, but it turned out it had psychiatric sequelae such as increased anxiety and increased suicidal thoughts and the like. So what's interesting is MIRA does not hit that site that is a low affinity site that requires high doses. So we are a partial agonist antagonist. So rather than do this U shape, we just go up and then, in a sigmoid, typical sigmoid shape, go up and then max out. And that turns out to be really good because we think that this inverted U shape accounts, and this inverse agonist like rimonabant accounts for why there's increased anxiety with THC for many people at high doses, and why there's psychosis, at high doses for some people. And since we don't hit that, we were not surprised to see that, for instance, mice don't get catalepsy, which is an animal model of psychosis, whereas they do with THC. But what was interesting is when we looked at low doses of THC versus MIRA, MIRA had no cognitively impairing effect at low doses, but THC did. But when we looked at high doses, we saw something really remarkable that in retrospect, when we went back and looked carefully at the literature, it wasn't, it was something that we should have, anticipated to some extent, which is that we saw that MIRA 1A not only does not impair cognition, it doubles the ability of mice 24 hours later to demonstrate their ability to learn and associate what they've learned and demonstrate it the next day. And what's really sort of impressive to us is they doubled their learning and memory the next day, but they weren't treated on day one when they were trained. So only on day two, when we tested them for their learning and memory, 30 minutes before the test, did they receive the drug? And that's a big deal for two reasons. One is it means that compared to the placebo group, they had exactly the same bit of information and knowledge. So that's like saying for those people who are mildly demented and lost some of their knowledge, it may still be something that they can recover function in because they don't have to go back and relearn everything while taking the drug. It doubles the learning and memory and the ability to perform cognitive tasks when you get the drug. And of course, when the drug wore off, those effects wore off. It didn't make these mice smart in a permanent sort of way. It just improved their performance on learning and memory tasks. And the other thing that's worth noting is that this was normal mice. So however smart evolution saw fit to make mice, this makes them do twice as well in these performances of learning and memory. And that is also remarkable. We are exploring, understanding the mechanisms better, but the implication is that we could take people who have mild, maybe moderate dementia, treat them, and have them actually symptomatically treat them and have them recover memory and learning that they've done but just can't access in the way that they used to be able to, and restore function that has been lost. That's the ultimate goal here. And, from the mouse studies, that's encouraging. We are now moving forward to trying to do this in, the FDA doesn't want to approve things that are not for disease-related indications. So, making people perform better on tests is not one of the indications the FDA is interested in. And also, age-related declines in cognition, not interested in. Those are normal part of the process. So we really have to demonstrate in a dementia model that we can improve cognition. But, unfortunately, that takes a while to wait for Alzheimer's mice that will overexpress plaques and tangles to start to get cognitively impaired. And so we need to, wait that amount of time for them to get older to test it. But we're encouraged. The other thing, of course, is that because it isn't cognitively impairing, it's cognitively enhancing, that treating patients with dementia who quite often have, anxiety related to loss of memory and other age-related, increases risks of anxiety disorders. Unlike the treatments that are commonly used, Xanax, Valium, benzodiazepines, all of which do dramatically impair cognition in helping the anxiety, this would be a drug that could help the cognition and anxiety without having the burden of, the worsening cognition. So that's MIRA-1A. I'm going to pause for a chance for you. That's sort of where we're going, is trying to target early dementia, improve cognition with help with anxiety without worsening these effects that benzodiazepines have, and move forward. But let me pause and make sure I'm on the right track here. And
Absolutely. Yeah. I mean, it's very striking, very, very striking in terms of what this compound can do. And thank you so much for going into so much detail about it, because I think that's going to be very helpful and useful for our listeners and for me as well personally. I just love to hear about how these drugs, how these things work. And it's very interesting how you came upon making a better 2.0 version of THC by just removing that one carbon, and that was able to, because I think, yes, the focus is usually in the space is to how to deliver THC better, increase its bioavailability, but to have a compound that's a 2.0 version of it, I think that's dramatic, and that's very, very exciting. So
Yeah, thanks. And there's a whole broader discussion, which
mhm.
I'm not suggesting we get into, but I think a lot of the real advantage that patients get from medical marijuana is that they can go and choose something that they know what it's doing. In other words, they go, they talk to someone who is an expert in the use of these drugs, and they'll say, well, for your particular symptoms, this is the variety that we recommend. And they're not FDA regulated, and therefore they're not consistent in what you get, but never underestimate the power of placebo in combination with some
Right.
therapeutic benefit. And I think because doctors spend less and less time with patients talking to them and don't educate them about what their symptoms mean, what the disease is, and how the drugs work, being able to go and choose themselves and play an active role in their health, I think is a big deal. We're not going to target those people, who are doing this, because that's what they want. But what we hope is to have that knowledge and independence and the ability to choose. And they've had bad experiences with side effects of FDA-approved drugs. But what we're trying to do is for those people who want the insurance to cover their drug, the FDA to regulate it, so you know exactly what you're getting is the real thing at the right dose and purity, and to have the benefits that this drug provides and minimize the side effects. That's really what we're aiming for is that area. And I will tell you, at Hopkins, we can't prescribe any of these medical marijuana because they're not prescription drugs, right? You use a card. It all sort of routes around the FDA.
Yeah.
But because it's a Schedule I drug, Hopkins will not let us write prescriptions or not let us dispense it for patients. So this overcomes a lot of those impediments for people to get
Absolutely
to a cannabinoid that, like you say, I like the way you put it, is a 2.0 drug to try to maximize benefits and minimize side effects.
Absolutely. And I think people do get caught up in the benefits, but once reality hits with the side effects, I think it's kind of like, okay, where do we go from here? And that's exactly what you're addressing. So that's wonderful. And I think that it's so key. It's so true that walking into a doctor's office, I think patients really need to be dispensed that information to not everyone's a scientist, but I think to some degree, the patients need this kind of information in terms of what is this drug going to do, and how does it basically work, and what can it do for me? So that's wonderful. I really like those perspectives. Yeah, I could sit here listening to you all day talking about the receptors, and yeah, it's just taking me back to grad school. So love it. Okay, so do you want to move on to ketamine or?
Yeah, sure. So happy to move on to ketamine. So ketamine, as sort of you conveniently let me freestyle a little bit at the beginning, had a chance to do an intro of, one of the great needs is that 1/3 of people who will not respond to multiple trials of antidepressants and still suffer from depression, which, as we've talked about, is life-threatening, not to mention the tremendous morbidity it is. In studies that had been done actually a while ago, it was depression was the second leading cause of disability of chronic illnesses, only to cardiac disease. But it was more debilitating than diabetes and pulmonary disease and kidney disease and those kinds of things. So it has a tremendous impact, not just on people who are affected, but their whole families and their network of friends and family. So having better ways of treating that came about when Janssen had what I think was somewhat heroic effort to try to come up with a treatment that would address this. They found that work that had been done previously at places like Yale had shown that low-dose ketamine is a drug that's used for anesthesia still in kids and particularly and pregnant women. It's on the list of WHO, top 100 drugs discovered by mankind because it happens to be a anesthetic that can be used in Africa where you don't have in the bush in Africa where you don't have access to intubate someone and put them on a respirator because it does not cause respiratory arrest like propofol does. Poor Michael Jackson died as a result of that.
Yeah.
Yeah. So this doesn't do that. So that's great at higher doses, but at much reduced doses, it was found that mice had a behavioral response to low doses that they do to all the antidepressants that have been tried, SSRIs and SNRIs, and those are drugs that affect serotonin and norepinephrine. They're reuptake inhibitors, so they increase the levels in the brain. And so it was tested in humans and found that single doses could last, even though it's an hour and a half, 2-hour half-life, the effects can last up to 7 days. They happen within hours to a day, not the 2 weeks, 1 to 2 weeks to even kick in, and then 4 to 6 weeks to maximize any particular dose level. So that was great. And Janssen went so far as to get approved Spravato, which is the left-handed molecule of ketamine. And they were able to deliver it intranasally. It has got poor bioavailability. So when you take it, maybe 20% of what you take will be available to affect the brain after the absorption is taken into account and then the metabolism. And so that was great. They were able to find that if you took it 2 times a week for a month, 1 time a week for another month, and then either tapered or perhaps continued, that's where the studies still need to be done. Upwards of 60% of people who have treatment-resistant depression will still have a response, and even a major response to this treatment, which is great. Unfortunately, the difficulty is because it's ketamine, as some people will know, it's a club drug that is used by some for its dissociative / hallucinatory side effects, where people sort of describe, looking down on themselves or being in a dreamlike state and what have you. And so therefore, in getting approval for the use, even though it was a low dose for an approved drug, the FDA required a REMS program to manage the risk associated with it. And so the decisions were made, and I'm not going to get into the details, I don't know them, but the decisions were made to make this intranasal treatment delivered by the patient. But then because of the REMS program, clinicians need to observe the patients for 2 hours. There is the risk of sedation, the risk of blood pressure changes, and then these dissociative side effects that 40% to 60% of people will get. But, maybe half people won't get those side effects. But that creates a huge problem because doctors have to be specifically trained in the use of this. The pharmacies that dispense it have to be trained in the use of it in the REMS program. And then insurance will just not pay much of anything. I think Medicare, we were told at Hopkins, would cover $15 to $20 is what they pay for a clinician to be there and observe a patient for 2 hours to make sure that they didn't have a bad response to the drug. we've done 1,000, or maybe thousands of doses of the drug at Hopkins in the Spravato clinic or in the studies that we participated in, and we never had a patient have a significantly bad outcome. Some patients became dysphoric and then were glad when it wore off, but nothing that required hospitalization or anything like that. And but so really, it's the limitations in using a drug that you have to go into the hospital or the doctor's office. You have to stay there for 2 hours. You can't drive for the rest of the day. And for the clinicians to dispense it, they have to go through a whole training, and then it has all of these requirements. And the insurance companies, are quite reticent to allow people to do it. It costs $1,000 a dose, say $800 for the drug, and then a couple $100 for the visit and all of that. So it is an expensive treatment. And really, if we could come up with a better way, safer way that wouldn't have these side effects, then that would be a great benefit. Ketamine is of particular interest to us for two main reasons. One is that it has a much better bioavailability. Oh, I should say, not shockingly, I think at this point in the conversation, it's worth mentioning that ketamine has two 6-member rings, one of which is converted to a 5-member ring. So this conversion is the same thing. It's not the benzene-like structure of the ring with, double bonds in it. But the other end of the molecule, the 6-member ring, becomes a 5-member ring. The first thing that we noticed is it dramatically changed the bioavailability in a KCO2 model, which is, these are human immortalized cells from the intestine. They form a monolayer, and when you put the drug into the top chamber, you see how much gets through that intestinal lining to the bottom chamber. You also measure how much from the bottom chamber gets pumped back out with the P-glycoprotein that's there to try to get xeno drugs, these foreign drugs pumped back out. And what we found early on was that, and we were very pleased with this, is that in terms of the intestinal absorption, and by the way, this KCO2 model has a very good correlation with intestinal absorption when it's been studied. And so what we found is that compared to the positive control, which was propranolol, an oral beta blocker used for blood pressure, sometimes people use it for anxiety, like violin players and the like, just acutely, but it's a beta blocker, and that's a positive control. Ketamine is not absorbed that well, 80% better absorption of ketamine. With the KCO2 model, we first of all found that we had better absorption with ketamine than even propranolol, 80% better even than ketamine. Ironically, more ketamine was pumped back out in this model than was pumped in, or that rather than was absorbed in. And so it's sort of hard to say how much better we were, because it was negative for ketamine, and it was not only did ketamine have good absorption, it beat the positive control of propranolol. So if you, sort of do the numbers based on saying, gee, if this is 20% oral bioavailability for ketamine, given, after you do the adjustments for the difference between ketamine and ketamine, we'd have something more like a 60 or 80% bioavailability. Now, that's just an estimate, because there are, in addition to absorption, you also have to, worry about metabolism. The reason why we're not as worried about that is there's some good studies that suggest that it's actually the metabolites have many of the active properties that have the antidepressant effects. So if that's the case, then even if there is metabolism, it won't affect the fact that we're absorbed much quicker. And then whatever the metabolism, if it then creates metabolites that are active, not a big problem. We'll still have better bioavailability. So that's in one. The second big deal is that we don't affect the same receptors with ketamine that ketamine does. The one that I can just focus on for the purposes of this discussion is we don't have opioid receptor agonist activity. And that's a big deal because much of the worry about addiction and the like with any drug will relate to its opiate receptor activity. There's even a bigger issue in the field that there are some who believe that it's the agonist activity of ketamine that accounts for the mood elevating effects of the drug. A lot of us don't buy this because even though there is data that is misinterpreted, in my opinion, that says, gee, if you throw in if you throw in naltrexone, which blocks opiate receptors, you block the antidepressant effects of ketamine. So that shows that it's necessary, but it's been shown it's not sufficient. So, and in this case, what's necessary is that you don't throw in naltrexone because naltrexone has effects on the opiate receptors that actually interfere with the antidepressant signaling that ketamine does. The fact that we don't hit opiate receptors at all helps to clarify the fact that since we still see antidepressant effects of our drug, and this is sort of hot off the press, since we still see effects of our drug, which we are in the process of, confirming in larger repeat trials, that means that it is not necessary for the antidepressant effects to have opiate receptor activity of the drug itself. It's really that naltrexone is doing this mischievous thing in the signaling pathway. And if we can get rid of side effects such as the addictive properties of taking a mu opiate receptor agonist, and it's orally available, has fewer side effects. I'm not going to get into the other receptors, but they also may mediate some of the side effects like sedation and the like, and blood pressure changes. So we are moving towards trying to see if we can create a drug that could be taken at home, safer, effective, orally available. That will really be a game changer in terms of treating patients who have severe depression and don't have the time or the money to come into the hospital and spend 2 days a week where they basically can't drive and have to spend 2 hours in the hospital just after they've dosed the drug, just waiting for it to wear off.
Indeed. And I think really the takeaway from the drug development approach, it seems to me at MIRA, is really to develop compounds that are a better version of existing, THC-based compounds, and that really minimize side effects. And I think that's huge. So that's, definitely would be game changing in this space. And I'd just like to ask, in terms of looking ahead, what other potential applications or developments do you foresee for THC-based compounds, analogs? And then I think part two of my question is also looking at the landscape of cannabinoid research and how far do you think it's come and how do you think it's evolving?
So let me take the latter part first, which is I think that, having a Schedule I drug has really slowed everything down in terms of research. It has been painful for us, for instance, some of the companies, Eurofins has a group in Taiwan, and getting THC as the benchmark to Taiwan was like trying to sneak, a shipment of drugs into the country. And that just, indicated to us how difficult it's been to do research in this area. So I think it's important to note that not as much research has been done because of the restrictions in terms of it being a Schedule 1. It is still a Schedule 1 drug, even though if you put it in sesame oil and you know, mix it in the way that the companies do that make dronabinol, it's Schedule 3. Still, isolated by itself, THC is Schedule 1. So doing research on it is still extremely difficult. I think what's good is that everybody's moving forward with trying to do the research. Let's find out how impaired people are in driving. Work is being done by Ryan Vandry at Johns Hopkins, a colleague and friend. He's got a fancy driving simulator to actually sort of look at this stuff. And this is stuff that, should have been done decades ago, but because it's a Schedule 1 drug, it is made it very difficult to do those studies. So I think the whole field is moving ahead with better studies. I think those studies will uncover not just the benefits, but also refine the risks. And I do think, that we can do better than nature has done in coming up with drugs that are less toxic. So I think more data to generate drugs that are better tolerated, better therapeutically, have benefits that we didn't anticipate, like cognitive improvement as opposed to impairment. And just the data that we get across the board is improving as people are able to do more research in cannabinoid space.
These are excellent insights, and I think the barriers. It's tough. For researchers like yourself in the field of dealing, it's not just a matter of dealing with any kind of a compound in a lab. You really have all of these regulatory barriers to contend with. But I think you're making great ground, and we need people like you in the space working at it. So, what are the short and long-term goals at MIRA? What can we expect in the coming months, years, and any insights
Yes.
into that?
So what we found out, the hard way is that it turns out nature doesn't make the 5-member ring versions of these because it's not straightforward how to synthesize. They're not easy to synthesize. So we are working hard. We have the ability to synthesize these drugs. What we need to do is, and with good purity, what we need to do is scale them up so that we can continue. So we are simultaneously, as we develop the ability to manufacture these, working with our colleagues at various manufacturing sites such as Recipharm and Benuvia and the like. We are working on getting larger and larger quantities for reasonable, costs. And simultaneously, we're working with Frontage that does all of the toxicity and tolerability and pharmacokinetic studies, and then the rat and dog studies that ultimately, once, it's a very prescribed set of studies, the IND enabling studies that one needs to do, and Frontage is doing this for us. So, we are getting them the drugs, they are doing the studies. Ultimately, we need to get a GMP quality, high quality for human use drug that we can then do the tests in animals. Then we do the phase 1 studies just to show safety in humans, and then the phase 2 studies where we'll begin to get efficacy, maybe some dose finding. And that's where that's where the money will meet the rubber will meet the road in terms of having a chance to see the antidepressant effects of ketamine and the pro-cognitive effects of and anti-anxiety effects of MIRA-1A. We are simultaneously working on, trying to improve even our 2.0 drugs, perhaps coming up with 3.0 versions. So we are actively and aggressively working to do that. So I wouldn't be surprised if we didn't get lucky and perhaps find an even better version than the versions we're working on now. We don't have anything to report at this time, but that is one of the other sort of to-do lists.
Excellent. Well, I Like to thank you so much for your time, Dr. Kaplin, for being on the show. This was such an insightful conversation. And I'm sure our audience are going to get a lot of important insights into the field and how companies like MIRA and how investigators like yourself are making huge strides in the area. And I only wish you all the best and looking forward to learning more about what comes of these two compounds and more from MIRA in the future. So I'll definitely be keeping my eyes peeled and staying tuned.
That's great. And again, thank you for the chance. We are passionate about our babies, but it's nice to get other people who find them of interest. And it's always good to get input. So thank you for your time and wonderful chance to discuss this with you.
Absolutely. Thank you very much once again, Dr. Kaplin. And that's the end of this episode of the X Talks Life Science podcast. If you liked today's show, don't forget to rate, review, and subscribe. Thank you, everyone, and see you all next week. Bye for now.
Thanks for listening to the X Talks Life Science podcast. If you enjoyed our discussions today, please share the episode with your friends and colleagues, and be sure to subscribe in order to be notified when a new episode is released. To join in on the discussion, you can find X Talks on social media, email podcast@xtalks.com, or comment on the articles directly. Links are in the show description. Take a moment to join our community at xtalks.com to get access to everything we have to offer, including webinars, job listings, virtual meetings, articles, and more. The views and opinions expressed in the podcast are those of the speakers sharing them. They should not be taken as professional advice and do not necessarily reflect the policy or position Honeycomb Worldwide. For further information, email us at podcast@xtalks.com. Thanks for joining us, and we'll see you next week.